All right, so moving on to chapter 12, which is all about antimicrobial drugs. Now, the ideal characteristic of any antimicrobial is to be selectively toxic. So what this means is the drug should be able to destroy the pathogen without harming the host cells. We're going to cover other important characteristics of antimicrobials. We will go over all of the different types of drug classes, how they work, what are their names, their particular grouping names, and also some specific details about whether they are naturally produced or synthetically produced in the lab. So other than being selectively toxic, hopefully they would kill the microbes rather than just inhibiting their growth. Also, we need to take into account that they will be administered to a patient and they will have to go from showing activity in a petri dish in vitro to being effective in vivo. So they must be able to function even when present in highly diluted form. So this would be as the result of metabolism throughout the human body. So hopefully they would remain potent long enough to act and they're not broken down or excreted prematurely. And hopefully they would not lead to the development of antibiotic or antimicrobial resistance, which unfortunately is inevitable and which we're seeing now is becoming a huge problem. Hopefully they also will be readily delivered to the site of infection, reasonably priced, and also not induce any allergic reactions, which again, this is hard to predict, and we always have a certain percentage of people that will develop an allergy to an antimicrobial drug. Now, some terms that you all should be familiar with. Prophylaxis, this would be using a drug to prevent infection. Antimicrobial, this is all inclusive for any type of antimicrobial drug, so any drug that will kill a pathogen that is causing disease. Antibiotics are naturally produced from different species of both fungi and bacteria, and the majority of antibiotics are used to treat bacterial infections. There are some exceptions, but please remember that majority will treat bacterial infections. And then these two terms here, synthetic, semi-synthetic, these drugs are chemically modified in the lab after being isolated from the original or natural source, whereas synthetic is completely synthesized within the laboratory environment. And then we have narrow versus broad spectrum, as you will see some drug classes or some specific drug names are classified as narrow spectrum, meaning that they are solely effective against a limited grouping of microbe types, specifically, for example, maybe only effective against gram-positive, versus broad spectrum drugs, which would more widely treat multiple types of infections, say those associated with having a gram-positive or a gram-negative cell wall. All right, so please remember, antibiotics, this term is referring to a group of drugs that are naturally produced. So they are naturally produced in, for the most part, a soil environment. So a lot of the Streptomyces bacteria and Bacillus bacteria, also mold. So the first fungus that we saw growing on a plate, it was a serendipitous discovery. Remember back to Fleming, saw penicillium growing on that agar plate and he saw clearing around bacteria. So penicillin was first discovered from that serendipitous experiment, and this is indeed a fungus. Also Cephalosporium. And why, you might be wondering, why would bacteria produce antibiotics to kill other bacteria? Well, the idea here is that by inhibiting other microbes in the same habitat, the bacteria or the fungi that produce those antibiotics have less competition for nutrients in space. So this is truly a form of natural selection and competition at its finest. Now remember, most important characteristic here of any antimicrobial, selective toxicity. It should target the pathogen without damaging the host cells. Now, as the characteristics of the pathogen become more similar to the host, so for example, a fungal infection in a human being, fungal infections are more difficult to treat and to attain this selective toxicity, especially if it's a systemic, say a lung infection, it's more difficult to cure, and because of this, usually more toxic side effects are seen. Now, we're going to go through all of the different drug classes, and I would like all of you to get the idea here of how these drugs are actually working. So when you are diagnosed with a bacterial infection and you are prescribed an antibiotic, 10 days later, a few days later, you feel better. How did that specific chemical drug actually target the pathogen in your body that was making you sick, kill that pathogen, and cure you of that infectious disease? Well, there are 5 different potential mechanisms of drug target action, one of which is inhibition of the cell wall, synthesis of that cell wall. Number 2, disruption of the cell membrane. So this would be the structure or the function of that membrane. Number 3, inhibiting the synthesis of nucleic acid. So a lot of antivirals will work this way. So remember, a virus targets the host cell and it hijacks that host cell's molecular machinery to make new copies of itself. So some drugs will actually act as base substitutes. They're called nucleoside substitutes, or they will actually stop one of the steps of viral replication along the way. So remember the stages of how a virus infects a cell: attachment, fusion, entry, uncoating, synthesis, assembly, release. A lot of times when we're talking about viral infections, this inhibition of nucleic acid synthesis is referring to just replication of the viral particle itself. Also, a lot of drugs will inhibit protein synthesis. So they will target different particular regions of the ribosome, either the large subunit or the small subunit, or the region, the interface in between the large and the small where it reads the mRNA. Or some drugs may actually block key metabolic pathways. For the most part, the ones that are mentioned in this chapter are metabolic pathways that are involved in folic acid synthesis. So you have these compounds that will act as a competitive inhibitor to specific enzymes, blocking the active site and preventing that enzyme from carrying out its catalytic activity. And because of that, the cell will die. So all of these, please remember, all of these are the 5 mechanisms of how antimicrobials actually work on the cellular level. Now here's a picture of it. So number 1, again, there's a group that targets how the cell wall is effectively synthesized. Another group will target the membrane. Another group targets the synthesis of nucleic acid, DNA, or RNA. Another group will target the ribosome, either the large or the small subunit, or it will target specific enzymes involved in the folic acid synthesis pathway. So the antibiotics that will target the synthesis of peptidoglycan will target the bacterial cell wall. And so these groups of drugs include penicillins, cephalosporins, and they will be active on young growing cells. Also, it's worthy to note that they are less effective against gram-negative bacteria, so there seems to be a higher incidence of antibiotic resistance with regards to gram-negative bacteria and the use of penicillin-like drugs. So again, it's active on young growing cells. So once the cell is exposed to cephalosporin, it will induce these weak points lacking peptidoglycan that should be synthesized, and this will compromise the integrity of the cell wall. So upon exposure to a hypotonic environment, the membrane bulges, the water will enter the cell, and the cell will lyse. Now, the major source of penicillin and its relative drugs are from Penicillium chrysogenum. So this is a fungus, and generally these antibiotics contain 3 chemical components. The most important or worthy to note here is the beta-lactam ring. So all antibiotics that contain this beta-lactam contain a highly reactive 3-carbon, 1-nitrogen ring, and they all work to target the synthesis of the cell wall, and actually half of them are beta-lactams. So penicillin G and V are the most important naturally occurring forms. Penicillin is indeed the drug of choice for a multitude of gram-positive streptococcal infections, also meningococcal, syphilis-associated infections. And then we have the semi-synthetic penicillins like ampicillin or amoxicillin, which have broader spectra of treatment. Also, there are some noted bacterial infections that have this associated penicillinase. So this is an enzyme that some bacteria devise to basically chew up the antibiotic penicillin. And so in that case, we would move to methicillin, nafcillin, or cloxacillin. Also, in some cases, people have allergies or resistance, so then we need to move to different types of drugs. Now, the cephalosporins will work via the same mechanism of action. They are isolated from another fungus, Cephalosporium, and they're relatively broad spectrum. They're resistant to most penicillinases and actually associated with fewer allergic reactions. Now, it's good to note that there are actually 4 generations of these drugs. So as you go from the first up to the 4th, each group is more effective against gram-negatives than the one before. And so this would be with an improved dosing schedule and also fewer noted side effects. Now, some additional beta-lactam drugs include the carbapenems and monobactams. So carbapenems can be used to treat a broad spectrum source of targets with aerobic and anaerobic pathogens, usually given at a low dose. We actually are starting to see, in the case of KPC, that's carbapenem-resistant Klebsiella, so we saw that in the video that I posted. Also monobactam, so this is more narrow spectrum drug for infections by gram-negative, and this would be aerobic bacilli, and also maybe used by people who are allergic to penicillin. Now, the non-beta-lactam cell wall inhibitors would include vancomycin, so this can be used to treat Staph aureus infections in cases of penicillin and methicillin, MRSA cases, or if the patient is allergic to penicillin. This is associated with restricted use, though, because of its associated toxicity, and it is hard to administer. And then we have another cell wall inhibitor, bacitracin. So this is usually seen in ointments, and this is produced by a strain of bacteria called Bacillus subtilis. Isoniazid, or INH, is in particular used to treat Mycobacterium tuberculosis cases. So this is the causative agent of TB or tuberculosis. Now, moving on to the second class of drugs as far as the mechanism of target, and that would be the cell membrane. So drugs like polymyxins are effective against some gram-negative bacteria, and these will basically poke holes in the cell membrane, similar to what we saw with surfactants and detergents for chapter 11, causing leakage, and will cause cell death as a result. Also, amphotericin B and nystatin, these work very well on fungal membranes, which again, same mechanism here. So this is just a cartoon image showing you on the top here. This would be for peptidoglycan, so weakening the links between the NAG and the NAM, and then also on the bottom here, polymyxin is working on the cell membrane rather than the cell wall. Now, that third class of drugs would be ones that target the synthesis of nucleic acid. So this would be the DNA or the RNA. Chloroquine, for example, will bind and cross-link the DNA double helix. Quinolones inhibit DNA helicases. So a DNA helicase basically acts as a zipper during DNA replication. So it is this molecular complex that will hop onto the DNA double helix and prior to the replication of each one of the template strands, it basically will unzip that double helix so that each template strand can be successfully copied and passed on to the next cell generation. So if the cell can't replicate, it will eventually die. And a lot of the antiviral drugs work via this mechanism. So again, think back to the basic goal or target of any virus is to hijack a host cell to make new copies of itself. So it wants to make a new copy of that genome, that viral genome. And some of these antiviral drugs will act as substitutes of the nucleic acid bases. So the A's, T's, C's, and G's make up DNA, which is a nucleic acid. And the larger of the two, the A's and the G's, are what we call purines. The T's and the C's are what we call pyrimidines. And so these antiviral drugs will act as substitutes mimicking the bases, and it tricks the cell into thinking that it's really the true DNA base, when in reality it's a fake, and so it actually causes the molecular machinery to stall at that particular location, preventing replication. Now also we have a ton of antimicrobial drugs that will block the synthesis of protein in cells. So there's thousands of proteins that are produced every second, and if the cell halts this process, the cell will die. So this is a particular mechanism of action with regards to treating infectious diseases that is perfect for bacterial infections because prokaryotic ribosomes are much smaller and completely unique in nature as compared to eukaryotic ribosomes. So they have this selective action against the prokaryotic ones. Now, aminoglycosides are one group of these protein synthesis inhibitors, such as streptomycin, gentamicin. And all of these protein synthesis drugs work in a different way to stop the translation process on the ribosome. So it's someplace on the ribosome, either the large subunit or the small subunit, or the interface between them. So the aminoglycoside will insert on sites on the 30S subunit and cause misreading of the messenger RNA. The tetracyclines will block attachment of the tRNA on the A acceptor site. So this is actually where the tRNA comes in on the ribosome before the protein, the amino acid is added. So these are different, just showing you a cartoon image of, for example, for the aminoglycosides, remember I said each one of these binds to a different spot on the ribosome. So aminoglycosides causes the mRNA to be misread. Chloramphenicol will actually stop the formation of the peptide bond. We also have other drugs here, tetracycline, for example, we just went over, blocks the tRNA. Erythromycin will actually prevent this ribosome from translocating. So when it's reading along the mRNA, it will translocate, it'll move one subunit at a time from left to right. So they all have a different mechanism of action. Now, with the aminoglycosides, a lot of these are products of various species of Streptomyces and also Micronospora bacteria. Now, a lot of them are broad spectrum, and they're especially useful against aerobic gram-negative rods and specific gram-positive bacteria. So, for example, streptomycin has been used historically to treat tularemia, TB, also isolated cases of the plague. Gentamicin can be used against gram-negative rods, and some of the newer ones, tobramycin, and also amikacin can be used against gram-negative bacteria. Now for tetracycline, these are also broad spectrum. Terramycin is a similar one, doxycycline. The only thing with these, usually associated with a lot more side effects than some of the other antibiotics, especially issues with nausea and effects on the GI tract, or even sometimes people are prescribed these antibiotics for a long duration, for an extended period of time, even years, and that can cause associated side effects even to the bone marrow or changes in coloration of the teeth, for example. But these can be used to treat STDs, Rocky Mountain spotted fever, Lyme's disease, typhus, acne, and also some protozoal infections. Another protein synthesis inhibitor antimicrobial drug is chloramphenicol. So this one has been isolated from bacteria, Streptomyces venezuelae, and now the one that is available is no longer derived from its natural source, so it is chemically modified, but it will block peptide bond formation. And remember, this one actually is associated with a high toxicity level. So I mentioned bone marrow. This one can cause irreversible damage to the bone marrow, and it has been used to treat such diseases as typhoid fever, also the rickettsial and chlamydial infections, and even in the case of infected brain abscesses. Sticking with the protein synthesis inhibitors, we also have macrolides. So erythromycin will attach to the large subunit, and it's associated with fairly low toxicity, can be used to treat mycoplasmal pneumonia infections, legionellosis, chlamydia, pertussis, diphtheria, and also as a prophylactic preventative for infection prior to intestinal surgery, also even gonorrhea, syphilis, acne, and then there's some newer forms of these available as well. A few others here, for example, Zyvox has been used to treat cases of MRSA and also VRE, or vancomycin-resistant enterococcus. We also have clindamycin. This can be used to treat abdominal infections, or Ketec. This is a newer one that can be used as a follow-up for infections that are resistant to some of the other macrolides. So there are many different groups here, all within the protein synthesis inhibitor realm. This is a pretty large group. And then we have other drugs that can inhibit specific enzymes, for example, enzymes that are required to synthesize folic acid. So a lot of the sulfa drugs, sulfonamides, trimethoprim, will block enzymatic activity required for tetrahydrofolate synthesis. And folic acid is really a precursor for not only the production of DNA and RNA, but also protein. So this is a very important chemical compound in all cells. So you stop the synthesis of this and you will kill the cell. And these drugs typically work via this mechanism called competitive inhibition. So basically, every enzyme has an active site, which is the particular site or location within that protein enzyme that binds the substrate. And so without having the active site available for that substrate to bind, it cannot catalyze its chemical reaction. And so it will block that active site. The drug competes with the normal substrate, preventing its activity. And also, since we're on the topic of drugs here, it's worthy to note that some of these drugs together can have either an enhanced effect or an antagonistic effect where they cancel one another out. So in this case, it would be a synergistic effect or an additive effect achieved by multiple drugs working together, requiring a lower dose of each. So they would enhance one another's effects. So this is just the example here showing you for the sulfonamides, right? I mentioned that it will target an enzyme that's involved in the folic acid synthesis pathway. So in this pathway, PABA is converted to this DHA and then goes on to be converted to DHA, THA, which is folic acid, and then folic acid is a precursor to DNA, RNA, and amino acids. So one of the group of these enzymes, sulfonamides, inhibits the enzyme that converts PABA to DHA. Trimethoprim is the enzyme that will convert the DHF acid to THA, or folic acid. So they're basically halting the pathway at different steps along the way. Now, as far as the specific drugs here, so they can be used to treat everything from urinary tract infections to bacterial shigellosis or protozoan infections. Also, a silver derivative of a sulfa drug can be used to treat burns, preventative for infections of burns, eye infections, and then trimethoprim for UTIs, for example. And then I just wanted to touch on a little bit of some of the other types of antimicrobial drugs. So for all those that I just mentioned, targeting either the cell wall, the cell membrane, the ribosome, or enzymes, right, or synthesis of nucleic acid, that all of those will target usually bacterial pathogens. But we do have other groups of drugs here that are used to treat fungal infections, protozoan infections, or viral infections. So for some of the newer ones, actually, that I didn't just mention here, Cindercid, that's one that has been talked a lot about for treating staph infections, enterococcus, that can cause endocarditis. Daptomycin is effective against gram-positive. Also, for fungal infections, so this is the new class here. Remember that a fungus is a eukaryote, so it doesn't have as many differences structurally and functionally to exploit or to target as compared to a prokaryotic cell. So a drug that is toxic to fungal cells typically is also toxic to human cells because they're both eukaryotes. But we do have groups of drugs that work pretty well, especially in the case of topical treatments. The systemic treatments or infections that are targeting, for example, respiratory lung infections are way more difficult to treat. But nystatin, amphotericin B, works by mimicking the lipids in the cell membrane of typical fungal pathogens and works pretty well, especially for topical treatment here and some systemic infections. Griseofulvin is effective at treating some dermatophyte infections, but is associated with nephrotoxicity, so this would be the kidneys. And then we have some azoles, which are more broad spectrum. Flucytosine, which can be used to treat some of those stubborn systemic diseases, and then the echinocandins, which work to target the cell wall rather than the cell membrane. So capsofungin is an example of one of these. Now, we also have groups of drugs that can treat parasitic infections. So we have the anti-malarial drugs like quinine, which is the most popular. But again, if you guys watched the video, you know that malaria treatments, these current malarial treatments, sometimes are not effective and are associated with a lot of side effects. We just don't have great drugs to treat that disease. Flagyl is another one that can be used to treat some protozoal infections. Also even tetracycline. So I said that the majority of antibiotics are used to treat bacterial infections. There are some exceptions, and this would be one of those examples. Also, in the case of treating a parasitic worm infection or using an anti-helminthic drug, usually these drugs will, remember, you're treating a multicellular worm, a multicellular animal, so these drugs will immobilize or disintegrate or inhibit the metabolism of that worm. So pyrantel, niclosamide, will destroy the scolex that attaches to the target cells, or will paralyze or immobilize the worm, or even inhibit the function of the microtubules. Some of these azoles work in that way. Now, for the antiviral, generally antiviral drugs, just think of how a virus actually replicates itself. So what does it do? It attaches to the host cell. It will then fuse with the host cell. It will enter that cell, right, uncoat, and start to synthesize new copies of itself and then release from the cell. So if you remember all of the steps of viral replication, you know how antiviral drugs work because they will target any step of the way. So some will block penetration of the virus into the host cell. Others will block the synthesis stages. So they'll block transcription or translation of the nucleic acid. So there are some that work as those nucleotide analogs, the base substitutes, like a drug that can stop replication of herpes viruses, or even hemorrhagic fevers, which these we don't have cures for, but it will at least halt the replication cycle. Or in the case of HIV, we have a thymine analog. Or some of these drugs will prevent the maturation of the complete viral particle itself. So there's a lot of text on here, but just remember every step of the way, right? Attachment, penetration into the cell, and then the synthesis, right? The transcription, the translation, and also in the case of HIV, so HIV is a retrovirus and it contains RNA, and during its infectious cycle, it uses one of the enzymes that it carries with it, this reverse transcriptase, to turn the RNA into DNA. And then that DNA will integrate into the host cell's chromosome via this enzyme called integrase. And once it does that, this is why people who have been infected with HIV are forever infected, because once it gets integrated into your host chromosome, it stays there forever. So there are some antiretroviral drugs that will actually stop the activity of this reverse transcriptase enzyme, or integrase, for example. Now, specific drugs used to treat influenza, for example, amantadine, are restricted almost exclusively to influenza A, and it will prevent fusion of the virus with the membrane. Tamiflu is one you guys have probably heard of that will block neuraminidase. So think of a flu virus, H1N1. The H stands for hemagglutinin, the N stands for neuraminidase. So it's an enzyme that's involved in the attachment and the fusion of that virus with the host cell. So Tamiflu blocks that activity in the case of influenza A and B infections. And then anti-herpes drugs, they act as nucleotide analogs or substitutes that are incorporated into the growing viral DNA chain, so it halts replication. And also in the same mechanism, you can find in oral and topical treatments for oral and genital herpes, chickenpox, and shingles. So these are actually all caused by different herpes viruses. And going back to what I said about HIV, so HIV is a retrovirus, and usually people who are infected with HIV are treated with a cocktail of drugs. So that cocktail of drugs are many different drugs that will each target a specific step of the retroviral replication cycle. So one, for example, will act as a thymine analog. One will target reverse transcriptase. One will target replication of the DNA, for example. So these are the two main ones, but usually there are other drugs that are involved in that cocktail. Now, also worthy to note here is a naturally occurring form of an antiviral component that our bodies make, and that would be called an interferon. So interferons are human-based glycoproteins, and our immune system produces them naturally. And the therapeutic benefits, so we have used interferons to treat specific viral diseases. Usually this will reduce healing time. It will prevent or reduce symptoms of cold and HPV infections. And also, surprisingly, interferons seem to slow the progress of certain cancers. Also, treatments of Hep C, genital warts, and Kaposi sarcoma, which is usually seen in late-stage HIV patients. Now, I wanted to bring up this concept of how antibiotic resistance evolves. So there's a few different ways in which bacteria can actually acquire these antibiotic resistance genes from their environment. And so I'm going to actually take this explanation from this microbiologist at the University of Texas, Carl Close, and he describes there's 3 different ways. So you're seeing here, it's detailed explanation. It says spontaneous mutations in critical chromosome genes or acquisitions of new genes. So yes, spontaneous mutations can occur. This is more of the rare side of cases. So the typical way in which antibiotic resistance is evolved or seen to evolve in bacteria is the acquisition of genes from the environment. And so there's three ways here. The first way would be what I'll call the funeral grab. So when bacteria die, they will actually consume genes in the environment, and they will go on to perpetuate those genes to the next generation. So that would be the funeral grab. There's also a mechanism through sex. So through bacterial conjugation, so this would be through the pilus, that the cells would form an actual bridge between them, and they would swap DNA, so they would actually acquire plasmids from the environment. So that would be the second way. And the third mechanism would be through what I'll call the viral pass. So this would be the bacterium would acquire these antibiotic resistance genes from a virus, which we know viruses that infect bacteria are called bacteriophage. So those are the three mechanisms. And as far as the molecular mechanisms, so once the bacteria actually acquire the antibiotic resistance gene, what is the mechanism by which that gene product is actually resulting in antibiotic resistance? So the gene that the bacteria acquire could code for an enzyme. I'll call this the ballistic missile defense. So basically those genes could code for enzymes that will shoot a missile at the antibiotic and chew it up. So like penicillinase. Or I'll call this mechanism the upchuck mechanism, whereby a gene could code for the ability of the cell to produce an efflux pump that would basically pump the drug out of the cell. Or through what I'll call stealth mode, that the cell could acquire a change in the target site. So for example, the antibiotics that stop protein synthesis could cause a minor change in the ribosome, the 30S or the 50S, which would result in the antibiotic no longer being effective. And another fourth way would be through mutation of an original enzyme. So this is kind of sticking with the theme of that stealth mode. So this is the cartoon image of all of the mechanisms that I just went over. So this ballistic missile defense, so penicillinase, the upchuck, right, with this efflux pump, and also the stealth mode, so you have the target site that is changed in a very subtle way. And so as far as the advent or the appearance of antibiotic resistance, this is to be expected. So the bacteria or any microbes, they want to live just like any type of cell that exists in the world wants to live. So what happens here is that if one of those cells actually acquires a resistance gene that allows it to survive in the face of an antibiotic, that will thrive on. It will replicate its DNA and be passed to future generations. And so eventually you'll go from one cell to a whole population of cells. And so typically what happens here is that there are certain activities that can encourage this process. So if you start off with a population of microbial cells, the majority being not drug resistant, but a couple that are drug resistant, when you expose those cells to a specific antibiotic, for example, if the individual does not take the whole prescription of that antibiotic, the course of the antibiotic, so they wait too long, or some of these cells are already there, what happens is all of the not drug resistant cells will be killed off, and you'll have the drug resistance ones that will prevail. And so they will start to replicate and remain over time to the point where now all of the cells are resistant. And it's estimated that 5% of all people taking antibiotics will experience a serious adverse reaction to the drug, side effects, or the this antibiotic resistance phenomenon. And sometimes what we see is disruption in the balance of the normal flora. So if you guys have heard of Clostridium difficile or C. diff infections, this happens when you're prescribed an antibiotic and it will kill off all of the good bacteria, leaving C. diff, for example, to overgrow. And so this can lead to a superinfection, and this is a big deal because C. diff can actually kill you. So it causes this incapacitating dysentery and diarrhea. And now we're seeing that using fecal transplants and the advent of fecal transplants to replenish the normal flora seems to be working very well. So there's some promise there. Now, as far as if you are diagnosed with an infectious disease, how does the doctor go about figuring out what to treat you with? Well, first he needs to deduce what you're actually infected with. And then there's multiple tests that can be conducted to determine what antibiotic those pathogens are sensitive to. Now remember, you could be infected with Staph aureus that 90% of those bacteria would respond to a cell wall inhibitor, penicillin or methicillin, but 10% of the strains of that particular species we're seeing antibiotic resistance. So it's very important to figure out what the profile of sensitivity is. Also determining whether the individual is immunocompromised. It's very important to identify the cause and then set up the sensitivity test. So the Kirby-Bauer disc sensitivity or disc diffusion test is the most common. So this would involve basically isolating the pathogen, culturing it in a lab, and then preparing an agar plate where you mix the agar with that culture, and then you would introduce these little paper discs soaked with a particular antibiotic and look around the disc to see if whatever is leaking out from or diffusing from this disc into the surrounding environment, if it would actually kill the bacteria in the plate. If it does, you would see this, what we call a zone of inhibition. The larger the zone of inhibition, the more effective the antibiotic. If you don't see any zone, you know that it's resistant. So this is a routine test that's done. Also, to look for the minimum inhibitory concentration. E-test is very similar. So this would determine the smallest concentration of drug that would inhibit that pathogen. And an antibiogram would be a profile of drug sensitivity. So this would allow the clinician to determine what we should actually treat the patient with. So like I mentioned from this image here, again, when you set up the Kirby-Bauer, you would actually look at the zone of inhibition around each antibiotic to determine which is the most effective. And the larger that zone, the more effective. This would be an image showing you that minimum inhibitory concentration test. So basically, you would, for each antibiotic, slowly increase the dosage and see at what particular dose do you see a lack of growth of that pathogen. So we see for lane 5 here, at whatever the dose is, that we see the pathogen is inhibited, right? So this is a positive growth control here. And then it takes a much higher amount of this drug to actually see inhibition. So this is good to determine actually how much of the drug is required, and also for which one would be effective as well. So remember, in vitro activity, what works in a petri dish may not necessarily work in a human. So this is kind of experimental. This is why it's called practicing medicine. Sometimes you don't know if these things are going to work. Now, as far as using antibiotics or antimicrobial drugs, it's best to choose a drug with the highest level of selectivity but lowest level of toxicity. And this is measured by what we call a therapeutic index. So this is the ratio of the dose of the drug that's toxic as compared to its minimum effective dose. And so therefore, a high index would be desirable. So that concludes chapter 12. I will see you guys next week.